Okay, so welcome back to this next video in which we are discussing protein folding and protein folding diseases. Okay, so we are in the process of discussing gain-of-function protein folding diseases, and the disease that we're now going to look at is the main example that is known of a prion disease. Okay, so we're going to look at prion disease. So let me firstly tell you what prion disease is all about. So prion disease is an infectious disease. It is contagious. You can catch it from another person who is infected. Okay, now it's unlike any other infectious disease, however, because the infectious agent that causes it is just a mere protein. Okay, and that's in fact where the name comes from. It comes originally from protein infection, okay, which was originally abbreviated down to proin. Okay, pro for protein and then I for infection, but then someone decided that proin didn't sound very good, okay, so we took it to prion. Okay, so this is a disease that is infectious and is caused by a mere protein. Okay, now we have found more and more, and we keep finding more and more proteins that are capable of prion-like behavior. Okay, but the main example of a prion disease that still stands as really the only proper notable example is mad cow disease, which is also, well, the proper name for which is bovine, which means pertaining to cows, spongiform, which means like a sponge, and then encephalopathy. Okay, and it's called that because it involves neurodegeneration of the brain, okay, and the brain gets holes in it basically and therefore becomes sponge-like. So it's a spongiform encephalopathy. Okay, now for short, bovine spongiform encephalopathy is abbreviated to BSE, and more colloquially it's just known as mad cow disease. Okay, because as the brain degenerates, the cow's behavior becomes more and more erratic. Okay, so mad cow disease. Right, okay, so let's discuss then the protein, the prion protein that underlies bovine spongiform encephalopathy. Okay, so this protein is so important in the field of prion disease that it is called the prion protein, or the major prion protein, and it's just abbreviated down to PRP. PR for prion and then P for protein. Now the prion protein is actually expressed all over the place in the cow's body and is believed to be also expressed in all over the place in the human body and also in the body of many other animals as well. Okay, so humans actually have the analogous protein that causes mad cow disease. Okay, and indeed it causes a similar disease in humans, it's just that disease is much, much rarer. Okay, so this is known as the prion protein and it's expressed all over the body and we don't know what its function is yet. Okay, however, the expression is highest within the brain, and this is why the brain is particularly at risk of getting the prion infection. Okay, so what happens then in bovine spongiform encephalopathy? Well, basically it's all about this protein. This protein can have two separate conformations. Okay, it can have the normal healthy conformation, which is called the PRPC. Okay, so you put this subscript C for cellular, okay, or cytoplasmic, and that's the normal conformation. So this is the protein nicely folded up in the normal way, and I might just draw a nicely normally folded up protein there. Okay, so here's our protein folded up in the normal way. That's supposed to be an alpha helix. Okay, however, the protein can also be in a really bad conformation, which is known as the PRP-SC conformation. Now, why is it called SC? Well, this is for scrapie, which is the analogous disease that is caused in sheep by this misfolded prion protein. Okay, so as I say, we have this protein in many different animals. It's in cows, it's in sheep, it's in humans, and it causes the analogous disease in all of them, and this disease is called different things in the different animals. So in cows it's called mad cow disease or bovine spongiform encephalopathy. In sheep it's called scrapie. Okay, so that's why that's denoted SC up there. okay? So the protein is now in a really bad conformation, a really wild conformation, so I might just draw it like that, just to denote a really wild conformation. Okay, now, what then is this protein in this awful conformation capable of doing? Well, this is the incredible thing. Basically, if you inject one of these badly folded prion proteins into a tissue with loads of these well-folded prion proteins, what happens is this protein is going to go up to the properly folded prion proteins, and it causes them to change into this misfolded conformation. Okay, so prion proteins, the fundamental characteristic then that all prion proteins have is that they are transmissible. They have these two conformations, and one of the conformations here is then transmissible. Okay, so it can go along to the protein in the healthy conformation and convert it into the bad conformation, and therefore basically the growth is just exponential. Proteins will bang into each other and they'll all gradually be converted into this bad conformation. And then of course the story is clear after that. These badly folded proteins then start aggregating, okay, into aggregates, and these form depositions within the brain, which then leads to neurodegeneration. Okay, and the aggregates that they specifically form are again amyloid fibrils. So these proteins can form amyloid fibrils. So you'll get amyloid plaques forming within the brain that will cause the neurodegeneration of mad cow disease. Okay, so how is this disease transmitted between individuals? Well, this is the sad story of what happened in mad cow disease. Okay, so in hindsight, the farming industry made a big mistake. Okay, the idea was simple. Our cows, how can we make them grow faster? Okay, well, at the moment they're just digesting grass, and grass is very difficult to digest. It's terrible to digest for humans, but for cows it's better to digest, but it's still, you know, a difficult way of getting nutrients. Okay, why don't we supplement their diet with huge amounts of protein? Okay, like gym goers supplement their diet with huge amounts of protein powders. Why don't we supplement the cows' diets with huge amounts of protein powders? Okay, well, where do we get the protein from that we're going to supplement the cows with? Well, we get it, unfortunately, from what's known as bone meal. Okay, and basically, bone meal involved grinding up the carcasses of cows which have been slaughtered for meat, okay, and then feeding that to the cows which were next, basically, okay, and all the portions of the cow that humans didn't want to eat went into the bone meal. Particularly importantly, the brain often isn't eaten in Western society, at least. So that went into the bone meal. So we were feeding cows other cows, and this bone meal contained the brains of those other cows. Okay, so if one cow had misfolded prion protein within its brain, this would end up in the bone meal, and then the other cows would be eating the misfolded prion protein, and then that's a way of transmitting this, okay, because then that can go into the other cows, the cows that eat the bone meal's body, and then those cows can end up with the mad cow disease as well, and that's how mad cow's disease started getting spread. Okay, right, so let's now talk about the analogous thing that can occur in humans, okay? So I should just summarize this up then. The amyloid fibrils that form particularly in the brain because of the high expression of the prion protein in the brain, which is why you're particularly at risk of getting the amyloid deposition badly in the brain, okay? That then causes the neurodegeneration which underlies the cow going mad. Okay, right. Now, the analogous disease in humans is extremely rare, okay? It's called Kruetzfeldt-Jakob disease. So, Kruetzfeldt-Jakob disease, okay? And as I say, it is extremely rare because cannibalism has at least been annihilated in the modern world. Okay, we haven't achieved world peace yet, but at least we have got rid of cannibalism. Okay, so Kruetzfeldt-Jakob disease, it's the exact same protein. It's this exact same prion protein in humans that can misfold in humans and again can form amyloid depositions particularly badly in the brain because it's most expressed in the brain. Okay, right. So how do you actually get Creutzfeldt-Jakob disease? Because we don't, you can't, how do you catch it? Well, most cases of Creutzfeldt-Jakob disease are inherited. Okay, they're familial. Now, how can this be familial? Well, if you get mutations in the prion protein that make the prion protein more likely to misfold into this other conformation, then you're going to get one prion protein maybe in your body turning into this bad conformation and then transmitting that bad conformation to all the other prion proteins, and then that causes the Creutzfeldt-Jakob disease. Okay, so usually it's caused by mutations in the prion protein. Okay, ways that it can be transmitted then, there are cases of where it has actually been transmitted to someone who hasn't got a mutation in the prion protein. Now, how can this occur? Okay, well, in the cows it was being transmitted because the cows were eating other cows' brains. Okay, we don't do that in human society. So how does it occur? Well, basically you need to have parts of someone else's brain getting into you. Now, there are examples of how that can happen, okay, in medicine, okay, because people with human growth hormone deficiency used to be treated by human growth hormone replacement. Now, before the rise of recombinant DNA technology, where did we get the human growth hormone from? Well, we went to dead people's corpses and we took the human growth hormone from their anterior pituitary gland. Okay, so we harvested human growth hormone from the brains of people who have died. And then that was being injected into people with human growth hormone deficiency. Now, if you took the human growth hormone from someone's brain who was suffering with Creutzfeldt-Jakob disease, then it would have these misfolded prion proteins in, and then you could give the person who was receiving the whole human growth hormone injection Creutzfeldt-Jakob disease. So that's a example of where this can actually be transmitted between humans, but it is very, very rare that it's transmitted between humans. The other concern that is still controversial as to whether it's true is as to whether mad cow disease can be transmitted to humans. So, for instance, if humans eat cow brain and the cow had mad cow disease, and we get cow misfolded prion protein into us, could that cause our prion proteins to misfold and therefore give us Creutzfeldt-Jakob disease? Okay, that's still controversial as to whether that exists, okay? The evidence is very low for it. Okay, so that's the prion diseases then now. Let's turn our attention to the final protein misfolding disease that I want to talk about, which is an example of a serpinopathy. Okay, now just like the lysosomal storage disorders, the serpinopathies are a large, well, I should have serpinopathies. They're a large group of diseases, okay, but we're just going to discuss an example of a serpinopathy. Okay, so firstly, let me tell you about serpinopathies in general, however. So, what is a serpin is the first thing we need to ask. Well, a serpin is a serine protease inhibitor. So the SER is for serine, the amino acid serine. The P is for protease, okay, and then the IN is for inhibitor. Okay, so these are proteins which inhibit serine proteases. Okay, right. Now, basically in serpinopathies, what happens is you get a mutation in a serpin, in one of these serine protease inhibitors, which causes it to misfold. Okay, so you start with a mutation, okay, which causes the protein to now misfold, so the serpin misfolds, and then what happens is this serpin, which is misfolded, starts to aggregate. Okay, now they don't form amyloid fibrils. This is one where it isn't an amyloid aggregate. Instead, what they form is what's known as ordered polymers. Okay, which is not quite an amorphous aggregate, it's more ordered than that. Okay, so let me just draw a little picture of this. So basically the idea is if we represent a misfolded protein as a little blob here, what they're going to do is they're going to just form a huge great string basically, and it is compared to just beads on a string. Okay, so these are the misfolded proteins in a huge great string like so, and this is what's meant by an ordered polymer. Loads of these proteins just bound together in a great big string like so. Okay, right. Now, this then is retained in the ER. Okay, so this ordered polymer remains in the endoplasmic reticulum. Okay, and then this ordered polymer within the endoplasmic reticulum of the cell which is making these serpins is then going to undergo cell death. Okay, so if I draw a picture of the cell here, so here's our cell, here's its nucleus, okay, and then here we'll say is the endoplasmic reticulum. What will end up happening is you'll get these ordered polymers of the serpins retained within the ER, okay, which is where the serpins are generally folded, okay, so they go to the ER for folding, and then of course these proteins misfold and then they form the ordered polymers within the ER, and these ordered polymers, you just can't clear them from the ER, and eventually it causes cell death. Okay, right, so why have I left serpinopathies right till the end? Well, the example of a serpinopathy that we're going to look at, which is alpha-1 antitrypsin deficiency, has components of both loss of function protein folding diseases and gain of function protein folding diseases. So the protein misfolds, and the pathology is caused not just by the gain of function, which is the fact that it forms these ordered polymers which then kills the cell which produces it, but also the loss of function of the healthy serpin, basically, the healthy alpha-1 antitrypsin. Okay, so it's got both loss of function and gain of function components to it. Okay, so we're going to look at alpha-1 antitrypsin deficiency. So alpha-1 antitrypsin is an example of a serpin. It inhibits the serine proteinase trypsin. Okay, and for short, alpha-1 antitrypsin is abbreviated to A1AT. A for alpha, 1 for 1, and then AT for antitrypsin. Okay, right, so let's think about what this is going to cause. So basically what happens is you get a mutation in your alpha-1 antitrypsin genes, okay, and this causes you to produce a protein that is going to misfold. Now, where is the alpha-1 antitrypsin protein actually produced? Well, it's produced in hepatocytes within the liver. Okay, so it's cells within the liver that are going to be producing the alpha-1 antitrypsin, and they usually secrete it into the blood. So if I show a hepatocyte here, usually what happens is the hepatocytes produce the alpha-1 antitrypsin, okay, it goes to the ER for folding, and then what should happen is it should be packaged along, going through the Golgi and then into a secretory vesicle, and then it should be secreted from the cell, okay, and it should end up in the blood, okay, and we'll discuss what its usual function is later on, okay, but for now, here is it in the blood. So what's going to happen then if you get a mutation in the gene for alpha-1 antitrypsin is that this protein is going to misfold in the ER, it's then going to form these ordered polymers in the ER of the hepatocytes, and that's going to cause hepatocytes to gradually die. Okay, so unfortunately what this causes is cirrhosis. Okay, so cirrhosis means the gradual degradation of the liver. It basically means liver cells gradually dying. Okay, so gradual degeneration of the liver. Gradual liver degeneration is what's meant by cirrhosis. And it's usually caused by alcoholism, okay, or by hepatitis. So certain viral infections of the liver can cause liver cells to die. Okay, right, so it's chronic liver degeneration. Okay, right, so that's one of the main symptoms of alpha-1 antitrypsin deficiency, and that's because of the gain of function activity of the misfolded alpha-1 antitrypsin. Okay, now let's think about the loss of function problems that are going to occur. Well, alpha-1 antitrypsin actually has an extremely important function, okay? It inhibits a serine protease known as elastase, okay? And this elastase, if it's not inhibited, what it ends up doing is breaking down the elastin that surrounds the alveoli, okay? So in the lungs, the alveolar spaces have got a lot of elastin, okay, which is a very elastic connective tissue. So these are the alveoli here. Now, in normal breathing, what happens is when you breathe in, air is brought in and the alveoli all bulge, basically. So they all get much bigger and fill up with air, Okay, then when you need to expel the air back out again, what has to happen is the elastic connective tissue of the alveoli has to then recoil back, and that helps you to expel air out. Okay, so for normal ventilation, which means bringing in fresh air and getting rid of the stale air, okay, you need this elastic tissue around the alveoli. Basically, if alpha-1 antitrypsin is deficient, elastase becomes far too active. It breaks down the elastin of the alveoli and they lose their elasticity, and then you have real problems expelling air in the expulsion phase of the ventilatory cycle. Okay, and this is what's known as emphysema. Okay, so emphysema is when you have real trouble ventilating because of the lack of elastic tissue within the alveoli. Okay, and it's usually caused by smoking, but this is an example of where it's caused by something other than smoking. Okay, so you get two diseases that are associated with alcoholism and smoking usually, without having done either of them. Okay, and in fact, if you do have alpha-1 antitrypsin deficiency, cigarette smoke is actually very dangerous for you if you inhale it, okay, because this adds to the degeneration of the elastin. Okay, so you need to avoid environmental pollutants, particularly cigarette smoke. Okay, now there are two main alleles for the alpha-1 antitrypsin deficiency found in humans. Okay, and they are called the M allele, that's the healthy one, and then the other variant that you can have is the Z allele. Okay, so those are the two main variants of the gene for alpha-1 antitrypsin. Okay, and it's the Z allele which causes the alpha-1 antitrypsin deficiency, and you actually don't need to have two of them to get the disease. Okay, so again, it's an autosomal disease, but you only actually need to have one Z allele in order to get the disease. If you have two Z alleles, then it will be more severe, okay? But even if you have one Z allele and one M allele, you will still get the symptoms of alpha-1 antitrypsin deficiency. Okay, right, so the emphysema that's caused by the alpha-1 antitrypsin deficiency, that's because of the loss of function of the alpha-1 antitrypsin when it misfolds, because you've got the Z allele of it, okay? And the cirrhosis, that's because of the gain of function of the misfolded protein, which forms these ordered polymers within the endoplasmic reticulum of the hepatocytes, and then leads to those hepatocytes dying. Okay, right, so that now concludes our discussion of protein folding and protein folding diseases. We have not covered all protein folding diseases that exist. There are a huge number of other ones that we haven't discussed. Okay, we've discussed a wide range, however.